Okay, so good morning, everyone. Whatever day you're watching this, good morning to you. So this is our 13th video teleconference turned recorded lecture for histology lecture. So if we take a look at our calendar here, we are now on the last month for this semester. So right now I'm recording this on November 29, Monday. So we have 3 lessons left before the end of this particular course. And those are the female reproductive system, the endocrine system, and special senses, organs of special senses. And that's before the final exam on the third week of December. So before anything else, I'll remind everyone again to please accomplish the interpreting the landscape portion of our module. Please do put your comments in about this micrograph. So this particular topic is about the male reproductive system, and our learning outcomes are to determine the tissues comprising each of the organs of the male reproductive system, to describe the histological organization of the testis and the seminiferous tubules, to determine the tissue changes in the seminiferous tubules during male gamete formation. So here's an overview of the male reproductive system. Okay, so you can see here how the passageway for the semen, no, seminal fluid, shares the same passageway for the urine. So, and that's the urethra. The main player here of the male reproductive system, what we call as the gonad, will be the testis. So it is here where meiosis happens from the onset of adolescence to the very later stages of the life of the male. As we know, males don't have menopause. Okay, so they continue producing sperm cells through meiosis throughout their life. So it's in this organ, the testis, where that happens. So we can see that the testis is actually composed of compartments here, separated by connective tissue. And in each compartment, we have what we call as the seminiferous tubules. So these are long coiled tubules. There are maybe 1,000 of them in one testis. And if you cut this tubule in cross section, you will have something like this. So this is the lumen of the seminiferous tubule, and these are the walls. It's on the walls where the process of meiosis happens for the production of sperm cells. So sperm cells are produced here. Once they are produced, no, or once we have the spermatozoa, they are not quite able to swim yet. So movement through the beating of cilia or through the passive movement of fluid from the seminiferous tubule to the tubuli recti, as we see here. So the tubuli recti is the one that connects this to the reti testis. Okay, will carry the spermatozoa into the reti testis, and from the reti testis, the spermatozoa will move passively through the ductuli efferentes, okay, efferent tubules to the epididymis. So it's in this epididymis where they actually gain their ability to swim. So they fully mature here. So this epididymis is a very long tube. And so if we unravel this coil, it's mentioned it can be around tens of centimeters, maybe 80 centimeters long. So can you imagine that wrapped in this comma-shaped structure that we see here? So on the bottom part of the epididymis here emerges a tube we call the ductus deferens, That's also known as the vas deferens. So the vas deferens is, it looks very similar to the ureter, from a distance. But if you look closely, as we'll see later on, the epithelial linings are quite different. So this is where the sperm will actually pass through during stimulation and sexual excitement, and this one leads to the prostate here. Okay, so where the vas deferens passes through the prostate, we have what we call as the ejaculatory duct. And in the ejaculatory duct, this is where several glands like the seminal vesicle, the prostate gland itself, actually contribute not to the consistency of the semen. And from there, the spermatic fluid, the semen, is going to move down the urethra, and here we can see another gland, the bulbourethral gland, which will contribute additional requirements for the semen. But actually this gland will sort of prime the urethra before the actual ejaculation. Okay, so that's the role of the bulbourethral gland. And of course here we have a longer urethra for the males than the female because it traverses through the penis, which is really a copulatory organ, and the main purpose of which is to transmit sperm cells into the female reproductive tract. So this one becomes erect during sexual stimulation, and that's with the help of what we call as cavernous structures inside, which actually fill with blood. Okay, so they're like sinusoids, but once they fill with blood, there's a mechanism wherein the blood cannot get out anymore, until that maintains the erection of the penis, and that lasts until, of course, after copulation. So these cavernous areas are divided into two main parts. We have the one that surrounds the urethra completely, and this is what we call as the corpus spongiosum. The corpus spongiosum is actually also seen here at the tip. So the glans penis is actually an extension of this corpus spongiosum. While here in the dorsal part of the penis, we have the corpora cavernosa. So there's a pair of them. Okay, so these three actually engorge with blood during sexual stimulation. So first, let's look at the structure of the testis. Okay, so here we have a section of the testis, and you can see a connective tissue here on the outside. This is the tunica albuginea. Okay, so this is sort of like the capsule, that wraps or protects the important parts inside the testis, and those important parts are mainly the seminiferous tubules. So we can see them here. There are many of them that are next to each other. So these are actually just connected to each other. Okay, they're just tubes that come to and fro. And so when we cut them cross-sectionally, we can see these cross-sections of them. So here's a closer look at the seminiferous tubule. So here's one. Okay. You can see there is a very thin interstitium. In the interstitium, you actually also have important cells like the interstitial cells of Leydig, okay, which produce testosterone. And then in the tubule itself, the lining is cuboidal and stratified. But this is a special type of lining because we have here the cells that will become sperm cells later on through meiosis. So there are several types of cells here in this lining, which we can see in the photo here at the bottom. Okay, so here is the lumen of the seminiferous tubule. And then here is the interstitium. And you can see in the interstitium there are smooth muscles that line the interstitium. These smooth muscles can continuously contract, so sort of like peristalsis, to help propel the sperm that's produced in the seminiferous tubule down to where they will be stored, which is in the epididymis. Okay, so if we take a look at the lining here, we can see several cells. The nourishing cells that will help nourish the sperm are the Sertoli cells, and they can be distinguished here through their nuclei, which are non-dividing. So it's very obvious they have a nucleus here with a very obvious nucleolus. So that means the nucleus is not dividing. Okay, so by that alone, you can distinguish that this one is a nucleus of a Sertoli cell. And then you have cells here that are near the bottom of, near the basement membrane. Okay, these are actually spermatogonia, which will later on undergo meiosis. And then here we have cells in the middle of meiosis, particularly prophase 1 of meiosis. And these are the primary spermatocytes. Okay, so these are the primary spermatocytes. And the secondary spermatocytes, as mentioned by the book, are much more difficult to come by because the phase where the sperm producing cell is in the secondary spermatocyte is so short. So they are often not seen in cross sections as we can see here. Instead, we have the spermatids directly. So we have the spermatids. And then the spermatids will mature, they will lose their cytoplasm, their nuclei will elongate. And here we have the late spermatids, which will mature and then which will become spermatozoan, which will break free out of the Sertoli cell. So this is actually the Sertoli cell holding the spermatids here. Okay, so this Sertoli cell is one tall cell. If I'm just gonna draw an arbitrary outline, that's gonna hold the late spermatids, and once the late spermatids have become spermatozoa, they will then break free of the Sertoli cell. So the Sertoli cell produces substances that will help nourish the forming sperm cells. By the way, the formation of sperm from spermatogonium to the spermatozoa is known as spermatogenesis. Okay, while the maturation of the sperm from the spermatids to a mature spermatozoa is called spermiogenesis. So here's an illustration of that, and you can see how the Sertoli cell is really a tall cell. Okay, and here you have the basement membrane and you have the myoid cells that will help in the peristaltic movement of the seminiferous tubule. It's a subtle peristaltic movement, so it's not like the gut. Okay, so it's very subtle. So the Sertoli cells are connected to each other here at the basal compartment through tight junctions. So here you have sort of the blood-testis barrier, and this prevents, the spillage or the crossing of sperm antigen into the blood of the male, and that prevents the formation of antibodies that will target the male's own sperm. Okay, so that's very important. The blood testis barrier here and these tight junctions formed by the basal portion of the Sertoli cells. And here next to the basement membrane, of course, you can see the spermatogonia. Okay, and these are still diploid, but once they divide, they start meiosis, they become the spermatocyte as we see here. These are the ones that were caught in early prophase 1 or late prophase 1. And then you have here the late spermatids. So again, in this illustration, the secondary spermatocyte is skipped. And here you have the late spermatids as the early spermatids lose their cytoplasm, a majority of it, and form the tail or the flagellum. And here you have the lumen. Okay, so again, the Sertoli cells function is they produce a substance that is rich in fructose, no sugar, which is going to be sort of the energy source for the developing spermatozoa or sperm cells. Now, the function of the Sertoli cell is sort of dictated by a hormone called the luteinizing hormone, or released from the anterior pituitary gland. Okay. So here we have more illustrations of the Sertoli cells, okay, with their nuclei and very obvious nucleoli in the lumen or lining the epithelium of the seminiferous tubule, while here in the interstitium you have the interstitial cells of Leydig. So they are these cells with obvious sort of vacuoles, okay, that are left inside there because of the processing when the preparation of this slide was done. The Leydig cells are stimulated by the luteinizing hormone. I think I was mistaken a while ago. The formation of sperm here in the lumen is stimulated by the follicle stimulating hormone. Okay, while the formation of testosterone by the Leydig cells is stimulated by the luteinizing hormone, both of which are released by the anterior pituitary gland. Okay, so the the Sertoli cells rather produce their fructose-rich secretions really independently of the follicle stimulating hormone. This one stimulates the starting of the formation of sperm, no, through meiosis. So from the seminiferous tubules, the sperm which are matured but not yet quite ready to swim will move passively into the retitestis. Okay, so this is how the retitestis would look like. So it's made up of these anastomosing hollows. Okay, and then from the retitestis, they are going to be moved passively again through the efferent ductules or the ductuli efferentes, and this is how they look like. So the scallop shape here is due to the alternating columnar cells with cilia and then columnar cells without cilia. So these are cross sections of the efferent ductules that we see here. So ultimately leading the sperm into the epididymis where they are going to be stored. Okay, so both of these, the retitestis, the tubuli recti, which leads from the seminiferous tubule to the retitestis, and then from the retitestis to the ductuli efferentes, these are all intratesticular genital ducts, meaning to say they are still inside the testicles. Okay, so aside from the seminiferous tubules, we have these three in the testicles. And ultimately, the sperm are gonna be stored outside of the testicles, okay, technically, and that's the epididymis. Okay, so this one is already part of the extratesticular genital duct. So as I mentioned a while ago, the epididymis is made up of this highly coiled tube, no, on this portion of the testicle. And here is a cross-section of that. And you can see, unlike in the seminiferous tubule, the cross-section of the epididymis will most likely contain spermatozoa. Because as I mentioned, this is where they are stored. Okay? The lining here will be pseudostratified with stereocilia. And I think that's very important because when the spermatozoa are not used, they can be stored here for several days. They will be reabsorbed, no, or resorbed back to the body. Okay, so they will be recycled, resorbed back, and then replenished with new spermatozoa. And the resorption is thanks to the presence of the stereocilia, no, lining the cells, epithelial cells of the epididymis, as we see here. So the book also mentions that the stereocilia are not really accurately named, okay, but for this reason, but for now we will just call them the stereocilia. So when the sperm cells are called upon, say when there's copulation that's happening, from the epididymis, they are going to be brought into the vas deferens, no, or the ductus deferens. So that's the tube that we see here connecting the epididymis to the prostate gland. So as I mentioned a while ago, this looks like a cross section of the ureter, a very thick muscular layer. Okay, so these smooth muscles will actually contract, no, with peristalsis during sexual stimulation. And we can see the lining. This is where it differs from the ureter. Instead of a urothelium, you have here again called a pseudocolumnar or pseudostratified columnar epithelium with stereocilia. Okay, so the vas deferens is going to go through the prostate gland as we see here. So there are two, of course, the left and the right. Okay, from the left and right epididymis. And this portion where they pass through the prostate gland is called the ejaculatory duct. And so here we have the prostate gland described as a walnut-sized organ. Here's a cross-section of the prostate gland. No? So you can see how the gland is sort of tubular, but then there are other components, no, beside the tubes. And the gland is divided into three layers. We have the mucosal glands, the one from the inside. Then we have the submucosal glands, no, color-coded here as green. And the main prostatic glands here on the outside. Okay, so the prostate gland actually produces substances that will help sort of liquefy the semen before it comes out, no, of the body. So that's the reason why this is very important, because semen has to be liquefied first before it enters the female reproductive tract. So the prostate gland also produces a prostate-specific antigen, which in certain circumstances, when there is an enlargement of the prostate, which is associated with old age. No? So males will usually really have that problem, whether they like it or not, whether we like it or not, at the later stages of our lives, wherein our prostate gland will actually enlarge, particularly the submucosal and the mucosal portion, and they will sort of pinch off the urethra so that you'll have difficulty passing your urine. And this is actually not permanent, and there are some medications that can be used to sort of ease the symptoms, and this will just you will just recover, no, without even thinking about it. So that's normal, and that's associated with the increase of the prostate-specific antigen that I mentioned a while ago. But for certain individuals where the enlargement of the prostate happens in the main prostatic glands here on the outside, it can actually progress to prostate cancer. Okay, so it can become malignant. And I think this is the most common type of cancer in males, no, that cause morbidity, cause death. So it's mostly on the main prostatic glands here on the outside. Also in late adults, the lumen of the prostate gland here would actually fill with concretions, sort of accumulation of the secretions of the organ, because there is an acidic portion of phosphatic acid, no, released by the prostate gland, and they sort of form crystals here. So in late adults, this will be filled with those crystals. So here's a bigger picture of that. And then we also have the seminal vesicle. So as we see here, so the seminal vesicle has this very elaborate folded lining as we see in these micrographs here. Seminal vesicle actually contributes, I think, to a majority of the volume of the semen that is ejaculated. They also secrete fructose-rich substance, which again maintains the health of the sperm cells even in the hostile environment of the female reproductive tract. And finally, we have the copulatory organ, no? The one that actually helps transmit the sperm cells from the gonads to the female reproductive tract, where eventually, hopefully, they will be able to find the egg and one of them will be able to fertilize it, and that's the penis, no? So, as I mentioned a while ago, the structure is composed of, so it's tubular. If you cut it cross-sectionally, you will find that in the dorsal portion, there will be two corpora cavernosa. Okay, so, and then at the bottom, completely surrounding the urethra will be the corpus spongiosum. So these are similar tissues. And here's a larger view of the corpus spongiosum. You can see the urethra here. You can see urethral glands of Litre. And you can see the trabeculae that fills with blood, as the penis actually is stimulated, and there's sexual stimulation. And this filling with blood actually sort of causes the closing of the blood vessel that leads blood out of the penis. Thus, the blood there stays, okay, and the penis stays erect throughout the sexual stimulation until ejaculation. So you can see the structure here. So this is, I think, a longitudinal section of the organ. Okay, so this one ends our my discussion on the male reproductive system. So just some reminders. Quiz number 9 on the male reproductive system will be on December 6 for 113YB and on December 7 for 113YA and YC, at these time slots. So there's a big chance that the next lecture will be a recorded one again. That will be on the female reproductive system. So I will be posting that one soon also. So thank you for listening, and have fun learning about the remaining topics on histology.